Today, we're diving into the incredible story of how computers went from being, glorified calculators to arguably the single most powerful tool we have in the search for new medicines.
Yeah, this quote really hits it home, doesn't it? Right now, it is almost impossible to imagine pharmaceutical research without computers at the center of it all. But it wasn't always this way, not by a long shot. We're going to explore exactly how we got from a world of pure trial-and-error chemistry to the data-driven design we see today.
So here's our game plan. First, we'll set the stage with the huge puzzle that is drug discovery. Then, we'll look at a couple of powerful statistical tools that scientists use to get a handle on all the uncertainty. After that, we're going to jump in a time machine to see how the tech evolved to make it all possible, and finally, we'll look at the massive impact it's had.
All right, so let's start with the puzzle itself. Before computers came along, trying to find a new drug was like navigating a maze with a near infinite number of paths. It was just this monumental task, a true test of creativity and, honestly, a lot of patience.
I mean, really think about that. Out of an almost endless sea of possible molecules, how on earth do you find the one that actually works? For decades, the only answer was a blend of cleverness, chemistry know-how, and just a ton of manual work in the lab. Scientists would make a compound, test it, see what happened, and use that result to guess what to try next. It was a slow, painstaking process.
So, to move this process from more of an art form to a hard science, researchers needed tools to deal with all that uncertainty. You just can't test everything. So how do you make confident decisions when you only have a little bit of data? Well, this is where statistics, and specifically something called confidence intervals, enter the picture.
Let's make this totally real. Imagine you're running a malaria control program. You've got 10,000 houses that need to be sprayed, but you have no idea what the exact surface area of every single one is. And measuring them all? That's just impossible. So how do you come up with a budget you can trust? You use a confidence interval. Now, this doesn't give you one single magic number. Instead, it gives you a smart, calculated range. By just measuring a small, manageable sample of houses, you can figure out a range where the true average for all 10,000 houses is very, very likely to be. It's a way of being really precise about how uncertain you are.
So here's how it works in practice. First, you take a random sample, let's say 100 houses, and you find the average area you need to spray is 24.2 square meters. Then, you use a statistical formula that takes into account your sample size and how much the numbers vary, and you can calculate that you're 95% sure the real average for all 10,000 houses is somewhere between 23 and 25.4 square meters.
Now, this chart is really cool because it shows you why this works. Each of those vertical lines is a confidence interval from a totally different sample of 100 houses. That dashed line in the middle, that's the actual true average we're looking for. And notice how almost all of our little ranges manage to cross that line. Only one of them, the one with the star, misses. That's exactly what 95% confidence means. The method just plain works 19 times out of 20.
So what's the bottom line? How does this help with the budget? Well, the health officials were smart. They took the upper end of that range, 25.4 square meters, just to be on the safe side. Because it's much better to have a little too much insecticide than to run out, right? And that led them to a solid, data-backed budget of 5,080 liters. A really smart decision made in a very uncertain world.
Okay, so confidence intervals help us nail down the uncertainty in our measurements. But what about the uncertainty inside our models? Like, once you've built a complex model of how a drug interacts with the body, how do you know which parts are the most important? That's where our next tool, sensitivity analysis, comes in.
The simplest way to think about this is like a what-if machine. You build a mathematical model of a system, let's say tumor growth. Then you just start tweaking the dials. What if this number goes up by 10%? What if this one goes down? Sensitivity analysis is what tells you which of those tweaks cause the biggest ripples in the final result. It basically tells you where to focus all your energy.
But just looking at the raw numbers can trick you. The chart on the left shows the absolute changes, right? But a variable might look really important just because it uses big numbers, like if you measured something in millimeters instead of meters. The real magic happens on the right. By scaling everything to the same standard, like a percentage, you get rid of all that noise. Now you can see the real story, which factor truly has the most leverage over time.
But of course, there's another layer to this. So far, we've been pretending we can change just one thing at a time. But in the real world, and definitely in the human body, everything's changing at once. This is where the more advanced methods come in. They let you see how one factor is behaving while all the other factors are also shifting around. It's like trying to understand a violinist's impact not during a solo, but in the middle of a full orchestra. This gives scientists the most realistic picture of what really matters.
Okay, so we've got these really powerful ideas, ways to manage uncertainty and ask, what if? But to actually run thousands of these what-if scenarios, or to model that whole orchestra of variables, you need a heck of a lot more than a pencil and paper. You need massive amounts of computing power. This is the story of how we built it.
This story pretty much unfolds over 4 key decades. The 60s, that's where the first seeds were planted. The 70s saw these ideas start to really gain a foothold. The 80s were just a decade of explosive growth. And then the 90s, well, that's when it all came to fruition and the whole thing really started to pay off.
So back in the 60s, drug discovery is still mostly trial and error. But you start seeing the first computational chemists appear in universities. And they did something crucial. They created the Quantum Chemistry Program Exchange, the QCPE, so they could finally share their software instead of everyone working alone. Then the 70s brought the hardware. Big mainframe computers start showing up in pharmaceutical companies, and for the very first time, we get central databases to store and share the 3D shapes of molecules.
A huge leap forward in thinking came with QSAR. This was a method to finally turn a chemist's intuition, that a molecule's structure determines what it does, into a predictive math equation. For the first time, you could use a computer to predict how active a brand new molecule might be before you even bothered to make it.
Then the 1980s brought this whole revolution right to the desktop with the IBM PC. You had user-friendly software that made it simple for any chemist to draw molecules, and molecular graphics that let you actually see and manipulate them in 3D. By the time the 90s rolled around, it all came together. Drugs designed with the help of computers started to hit the market, which proved this new approach was the real deal.
And this leads us to another game-changing technique, structure-based drug design. Thanks to those growing databases of 3D protein structures, scientists could finally see the lock, the biological target they wanted to hit. And with molecular graphics, they could digitally design the perfect key, a molecule that would fit into it and produce exactly the effect they wanted.
So, what's the result of this half-century of innovation? Well, the impact has been nothing short of transformative for medicine, and honestly for all of society.
You can just see the industry's commitment right here in the R&D spending. By the mid-2000s, pharma companies were investing tens of billions of dollars every single year, and a huge chunk of that was being driven by the sheer power and promise of these new computational tools. And this, right here, is why all of this matters. All the code, all the databases, all the algorithms, it all leads to this. The new medicines made possible by these techniques have had a direct, measurable, and profound benefit on human health. People are living longer, healthier lives because of this work.
So we went from a niche academic field to an absolutely essential part of modern medicine. The journey of computers in drug discovery has been just incredible. And it leaves us with this one fascinating question for the future. Now that we have these tools that are more powerful than we ever imagined, what monumental health challenges are we going to solve next? What cures are now within our reach?